Testing treatments. According to drug statistics, there are 11,901 small molecular drugs, 2,674 biotech drugs, 4,196 of approved drugs. I think this is the number that pharmacists need. And approved small molecules, 2,700, 132 nutraceutical drugs, 6,652 experimental drugs, and only 268 withdrawn drugs. The total number of drugs is 14,575. Needless to mention the four phases studies from 0 to 3 of the drug before entering the market for sale. It requires at least 1 decade for any drug to finish all of its studies and get the approval from a regulatory agency like the FDA in the United States and Canada, the Food and Drugs Agency. The double standard, sometimes is called the golden standard, is also very important for the study to be completely randomized, what we call RCT or randomized controlled trial. Okay, that's good, but you might say why I should care about all of this. Because today on the internet you can rate and review the drugs you've been prescribed to, and on the website like drugs.com, for instance, even if you are an ordinary patient, you can vote and see the votes and the comments of other patients like you, and in turn this will achieve what is called the democratization of drugs. Like here is the rating and review of the drug with warfarin or coumadin, the anticoagulant drug. And if a lot of people are reporting a drug, the drug should leave the market immediately, for it is bad and or dangerous to patients. Although unfortunately that's not what's happening, many stories like this did happen, like the story of Vioxx or rofecoxib, the non-steroidal anti-inflammatory drug which has been removed from the market because it was causing heart attacks and stroke attacks, or the story of Avandia or rosiglitazone, the anti-diabetic drug which again was increasing the risk of cardiovascular problems and heart attacks. These drugs really killed a lot of people. Then the company paid a little fine in court in millions of dollars after selling billions of dollars of it. And finally these drugs really left the market and has been withdrawn. But to the company what happened was only the cost of doing business. Another story about bad pharma is the story of Herceptin, a drug for breast cancer that has been released to the market by force before even accomplishing its clinical studies or its RCTs. What the company did at that time was actually very, very evil. First, they released a documentary after paying a woman with breast cancer, saying, I don't want to mention her name, that she has been cured after testing their drug on her. The drug was Herceptin, of course, purposefully to sell the drug without the FDA approval. And when the FDA did not give the company the approval, they paid other people, probably from the company acquaintances, to protest on their company doors, crying that some of their families are suffering breast cancer and saying that they want the drug now and they trust the drug because it's a potentially life-saving drug. And in this way the company got the approval without doing any clinical trial. Today the drug proved to be so bad and toxic and a big failure. By the way, vaccines also cannot enter the market before finishing long-term clinical trials or RCTs for at least 1 decade. Some doctors today are urging for a new type of clinical trials. Trials comparing different drugs together, like drugs of the same molecule but from different company, like original and generic drugs. Drugs of the same effect but of different derivatives, like comparing different cough suppressants or different contraceptive pills, or comparing sildenafil versus tadalafil for instance, or comparing different drugs very closely related in effect, like phentermine and ketamine. Both are used in anesthesia. The first is opioid based, the latter is sedative, or comparing caffeine versus stimulant drugs, or comparing muscle relaxants like carisoprazole versus local anesthetics like lidocaine, or comparing hydrocortisone versus diclofenac like Voltaren. Both are anti-inflammatory, but the first is steroidal, the second is non-steroidal. Finally, doing clinical trials onto a mix of drugs in order to witness the drug's interactions and contraindications. Pharmaceutical technology. Drug delivery is a pharmaceutical technique used for delivering drugs to the patient, each according to his situation, like pills, ointments, sprays, effervescents, eye drops, suppositories, injections, and transdermal patches. Drug delivery can offer different options for the doctor and the patient. For instance, we use sprays for asthma, ointments to treat skin conditions, and eye drops in ophthalmic conditions, and we use injections for a quick or immediate release of the treatment. And we use the transdermal patches, delivering the drug in a sustained or extended release of the drug, so the drug can last for hours or maybe weeks. This type of pharmaceutical technique has two options. First, the transdermal patch can be removed whenever the patient would like. Second is the idea for the drug to be released in small dosages and last for hours or maybe weeks, so the side effects will be much more calmer and smoother. Any drug can be formulated in a way to be delivered via the skin in a patch, like contraceptives, cardiovascular drugs, analgesics, and psychoactive drugs. Antidote is the molecule that reverses the effect of another. For instance, the Super Mario mushroom Amanita muscaria having muscarine can be reversed by the antimuscarinic drugs or anticholinergic drugs like atropine from the plant Atropa belladonna. The calabar beans are the antidotes of the curare plant, the plant that Native Americans were using for hunting animals. Biotechnology. In biotech, we only have two techniques. First, the recombinant technique, which is a technique that requires understanding of genes. The technique starts by changing the plasmid of a bacteria or yeast by introducing the desired gene that is responsible of producing the protein drug, and this process is called transformation. Then after the bacterial transformation, we should transform the sample into a bioreactor, and the bacteria will be fermented, and in turn it will give the desired protein, and then after purification we'll get the output, and with a very great amount. The best example of the recombinant technique is the production of insulin. Although there are a lot of drugs, including some types of vaccines, that are produced by this technique. The second technique is called monoclonal antibodies. In reality, this technique is not new at all, and it is done actually by introducing something like an antigen that triggers the immune response of an animal. Then the animal will produce antibodies that will be purified and used as drugs. The best example of it is the plasma antibodies of the rabies serum or the plasma antibodies of the tetanus bacteria. Other examples is the anti-D antibody, which is an injection used in pregnant women with O-negative blood type. But there is an issue in these drugs today, because these are marketed today to be used against cancer with a MAP suffix and against autoimmune disorders. And here is actually not helpful at all, in a way that doctors are really misled by this technique, which they don't understand at all, because monoclonal antibodies can be used in treating diseases with tissue deterioration, like autoimmune disorders like psoriasis or rheumatoid arthritis, or inflammatory bowel diseases, or even against macular degeneration or osteoporosis, etc. The argument here is like the argument of who came first, the chicken or the egg, or the argument of cause-effect, and I think here we need to be careful and see who came first. Then we understand why using these drugs is not doing well. When a deterioration or a degeneration of a tissue is happening, structural proteins will certainly be elevated, like the elevation of troponins in myocardial infarction, which is an effect of the disease, while beta amyloid proteins, on the other hand, are causing the degeneration of brain cells in Alzheimer's disease. Same thing for cancer. Oncologists need to understand the theory well before prescribing these drugs, like for instance using monoclonal antibodies for treating leukemia. In leukemia, the lymphocytes are sometimes abnormal, which means the cell might have surface antigens that are different than the normal lymphocytes. These antigens are called cluster of differentiations, or CDs, like the picture you see. And if the drug is studied well, usually by ELISA, then something like rituximab, which is a monoclonal antibody, might be useful for targeting these antigens with high specificity and lesser toxic side effects. And who knows, maybe the treatment will potentially be a life-saving drug for the children suffering the deadly disease of leukemia. Future goals in medicine. These are some of the diseases that I myself think that we are capable of curing or at least treating in the future, like psoriasis, which is a skin disease autoimmune with high genetic predispositions, malaria, hepatitis, Alzheimer's disease, and leukemia. And needless to say that so many diseases could be avoided in the future if we can one day apply eugenics. Unfortunately, today we are poisoning leukemia children exactly like when we were doing in history to patients before the discovery of antibiotics. And we are dissecting tumors sometimes without clear evidences, exactly like when we were doing amputations to gangrenes. Anyways, medical breakthroughs. These are dreams and sometimes are fallacies, and all are not for treating anything, like athletes doping, longevity, regenerative medicine, hibernation versus cryonics, endless euphoria, curing the paralyzed and the blind. Athletes doping. The theory of stamina says that fitness or stamina is the amount of red blood cells or the level of hemoglobin in blood that is responsible of picking up the oxygen from the air. So low levels of oxygen in high altitude could increase the hemoglobin level in the body naturally by gene expression. This is already what happened to Tibetans living in the Himalayas. Athletes can exploit this idea and try to train in high altitudes and in turn increase their amount of red blood cells naturally without using drugs or doping. Longevity. The longest human lifespan to be recorded is Jeanne Calment of France, 1875–1997. She lived up to 122 years, 164 days. In mammals, the bowhead whale is the longest that can live up to 200 years. The giant tortoise can live up to 300 years. In plants, it's been said that the Great Basin bristlecone pine can live up to 5,000 years. With these informations having in mind, some scientists are beginning to theorize a theory of longevity, which is called the theory of telomere. Telomeres are the repetitive nucleotide sequences at the end of a chromosome, and in humans it's TTAGGG with a complementary DNA strand of AATCCC. This elongation, they are saying, declines each year by the effect of an enzyme called telomerase, and they are saying that by using an inhibitor of this enzyme, the telomerase inhibitor, they might one day increase the longevity of the human race. Of course, this is certainly another dream and fallacy. But if you really wish to see yourself older or younger, for fun, try a photo editing app. Regenerative medicine. Regenerative medicine is a fake. The technology of stem cells is a big fallacy. This technology cannot and will never achieve anything of what they are saying, especially the idea of saying that we are capable of producing organs. In reality, what is happening today is a crime that need to be addressed in a court. Someone is selling his or her kidney for $2,000 and the other is buying it for $200,000. And I'm afraid that those doctors speaking regenerative medicine are themselves the criminals trading those organs in a way or another. The human body doesn't have this capability of regeneration. Our skin, heart, kidneys, brains, nerves, muscles, etc. are not regenerative. What is regenerative in our body are only the epidermis, the blood, bones, hairs, or fingernails. Although it's true that some plants and animals do have a regenerative capability, like salamanders. Here in the photo is the axolotl salamander from Mexico. This can regenerate an entire limb or tail even if it was completely cut off. Hibernation is sleeping for a long period of time, and it's already happening in nature. Like the dormouse can hibernate 6 months out of the year or even longer if the weather does not become warm enough. Contrary to cryonics, where the body is dead, and any company offer this kind of bullshit is somehow doing something like a modern mummification of the dead.